welcome everyone to our guest lecture presentation, our second one in this series. Wonderful to have you all here live with us, and welcome to those people who are catching up on the recording as well. Before I do make a formal start today, I did want to take a moment and acknowledge the traditional owners and custodians of the lands upon which we are all gathering here from New South Wales and Queensland. I myself am on Widjabul Waible country of the Bundjalung Nation, and I would like to acknowledge that this land always was and always will be Aboriginal land. And feel free to acknowledge your country in the chat if you would like to as well. So very exciting today to be welcoming our guest lecture presentation from Christine Jones. And I've just got a really short bio from your website, Christine, if that's okay. So Christine Jones is actually Dr. Christine Jones. She has her PhD and is an internationally renowned and highly respected ground cover and soils ecologist. Christine has a wealth of experience working with innovative landholders to implement regenerative land management practices that enhance biodiversity, increase biological activity, sequester carbon, activate soil nutrient cycles, restore water balance, improve productivity, and create new topsoil. Christine has organised and participated in workshops, field days, seminars and conferences throughout Australia, New Zealand, South Africa, Zimbabwe, Western Europe, Central America, USA and Canada, and has a strong publication and presentation record. I was very excited that we were actually able to grab Christine at this time to be able to present to us today. And on that note, I will stop talking and welcome you, Christine. Thank you so much.
Thank you, Simone. Thanks for that lovely introduction, and welcome to everybody that's on the session today. Can you hear me? All right, yep. Simone? Yep, okay, all good.
Yeah, perfect.
So today we've got about 60 minutes roughly for a formal presentation. I'm going to show you some slides and then around 30 minutes for Q&A, I understand. But that's sort of flexible. We'll see how we go. And I'm not really sure, because I don't know the backgrounds of the audience, other than you are interested in regen ag and then hopefully interested in the microbiome in the soil, as to which way to go. So I could even, maybe stop towards the end and say, do you want to go more down the horticultural path or look more at soil carbon or whatever. So we'll see how we go with the time. But the thing is that the soil microbiome is central to everything basically that happens in the soil, and we find that it's also central to a lot of other things that happen above ground as well. It's interesting when we start looking at microbiomes that the human microbiome has many in our gut. I'm talking about human gut microbiome. We have many of the very similar kinds of microbes in our gut as to what we find in the soil. So there's obviously been a long history of co-evolution between a whole lot of different species, and I guess we're just an assemblage of microbes anyway, in many ways, and an assemblage of organs that require microbial activity to function. And the soil is similar in many, many ways. We often don't think of it like that. We think we have similarities with our microbiomes have similarities with the microbiomes in animals, but we also have incredible similarities with the microbes in the soil. So the microbiome of all living things is very, very important. As we have become more aware in recent times, our human gut microbiome controls a very large part of our immunity, and there is also a very strong gut-brain connection as to, in other words, what is going on in your gut can influence your mood and your clarity of thinking and all those kinds of things. So there's been a lot of emphasis on microbiomes, but we haven't really focused down so much on the soil microbiome. So that's what I want to talk about today. And it's unfortunate that when we look back over the millions, if not billions of dollars that have been spent on scientific research into soils over the last century at least, and sometimes going back even further than that, they've all focused on inputs. Most of the research, not all of it, has focused on inputs rather than life in the soil. So we'll take a soil sample, send it away, get it analysed. It'll be subjected in a lab to chemicals that have no relevance really to soil. They have nothing to do with how soil functions. And then the lab will come up with some numbers saying, oh, guess what? You actually need to apply some nitrogen and some phosphorus and some calcium and magnesium and zinc and boron and copper and cobalt, whatever, based on this soil test, which really is not telling us anything about soil function, and the way the test has been conducted is not really relevant to how life in the soil works. So even in many organic systems, unfortunately, the emphasis has been on replacing chemical inputs with certified organic inputs. So we've still got very much this input-based production system, quite often based on soil tests and recommendations that come from soil tests. So in order to move forward from that, because that really hasn't got us very far in agriculture, we've realised that it costs farmers a lot of money for these inputs, that most of the inputs actually end up going somewhere else other than into our plants. So if we look at something like nitrogen, for example, around 10 to 40% is taken up by that plant, and that means 60 to 90% goes somewhere else. In Australia, that's mostly up into the air. But in some places like New Zealand and Ireland, it goes down into the water table and into the rivers and pollutes the water, and kills fish and other freshwater life. But also it's not very good for people to be drinking that water. So they spend millions of dollars putting fertiliser on and then millions of dollars taking nitrates out of the water. If we look at something else like phosphorus, which is very commonly used in Australia as an input, something between 10 and 15% is taken up by the plant, which means that, 85 to 90%, it doesn't, in this case, usually go somewhere else, but it becomes immobilised in the soil and cannot be taken up by plants once it's immobilised. So I could say the same about a whole lot of other inputs that we use, and they're not improving soil function, they're not improving the quality of our food, they're costing farmers a lot of money. So the question we've got to, which I think is basically like what Regen Ag is about, is restoring soil function. That's my definition of Regen Ag anyway. Doesn't matter how you do it, but if you're restoring soil function, then you are regenerating the resource base. So in order to do that, we actually do have to transition from using inputs as the basis of production to improving soil function as the basis of production. Or to put that another way, how can we transition from input-based production systems to process-based production systems? Because if we're going to get away from inputs, we actually have to understand what the processes are. You can't transition to a process-based system unless you actually understand how soil works. So the fundamentals for soil health is, number one, soil is a living system. It's very much alive, and it contains billions and billions of microbes, trillions in fact, and there's actually more diversity in the soil than there is in any other ecosystem on Earth. And the health of all living things is influenced by their microbiome. So we have to actually start to understand our soil microbiome and how it works. But what we've realised probably in the last decade or so is that one of the most important things about the microbiome is the diversity of microbes that make it up. So you could have a huge abundance of microbes of some kind, like just put out a whole lot of animal manure or something, and you're suddenly going to have a huge abundance of microbes, but it's not going to be a diversity of microbes. So our research has shown us that microbes work in teams, which we call consortia, and or consortia, maybe, I'm not really quite sure how to pronounce that, but they can't function on their own. So even some of our well-known microbes, like say Rhizosphere, that are in the nodules on legumes, Rhizosphere can't work alone. They have to have other bacteria working with them. Or if we look at something like a fungus like mycorrhizal fungi, which most of us working on soils would be very familiar with mycorrhizal fungi, but they can't function on their own. They have to have mycorrhizal helper bacteria and other kinds of microbes working with them in order to undertake their tasks. So a diversity of microbes is very, very important. And this diversity underpins most of the functions, or all of the functions really, that we want soil to perform for us. So we want nutrients to be available in soil so we don't have to go out and spend money putting them on. How do we get more nitrogen to be available, more phosphorus to be available, all the trace elements to be available? We want to have good soil structure. In other words, we want it to be well aggregated. We want to see all those little water-stable aggregates that make it porous and give it that beautiful crumb structure. because that is going to relate very closely to water holding capacity. And that's going to be important in a drought when we want soil to stay moister for longer. That's also going to be very important when we have an overabundance of water, as we have seen in recent years. We don't need to go there, but if soil is better structured, that water will actually infiltrate better. And we could do a lot about our boom bust situations with floods if we had water actually infiltrating where it falls rather than running off and gathering in rivers. So we need to have base flow into rivers, constant base flow rather than that boom bust situation. And resistance to pests and diseases, if I get time, I will cover a case study on that. And of course, depending on, I don't know whether people in the audience are actually crop producers or graziers, but our crop and pasture productivity is going to be influenced quite largely or immensely or almost totally by soil microbial diversity. And of course, carbon sequestration is related to all of those things above, and landscape function. So if we have reductions in soil microbial diversity, we get soil compaction, and that can happen anywhere. It doesn't necessarily have to be where we've got traffic from vehicles or traffic from animals. You could just go out into the bush somewhere where there's not enough microbial diversity and you'll find very compacted soils. So it's basically a lack of biological activity is what causes soil compaction, and we will have trace element deficiencies, and then people will be spending money again trying to remedy those with inputs, and all of these things lead to an increased need for inputs, and there will also be an inability to store soil carbon. So people who've registered for soil carbon projects, listen up, because microbial diversity is going to be the thing that will get you that carbon. So all the functions that we would like soil to perform are carried out by a wide diversity of microbes working together in teams. That's probably the most important slide of the whole thing. A wide diversity of microbes working together in teams. So where do these microbes come from? Now, if we were having a face-to-face discussion today, if I was actually able to be with you in person, I'd be asking you that question right now. And I'm just back from New Zealand, and whenever I asked that question, I was greeted by a whole lot of very blank faces and people coming up with answers like, whoa, well, you know, you could add some compost, or you could add this, that, or the other to actually increase microbial diversity. And the thing is that microbes have, the soil has a microbiome. So we have a soil microbiome. It has its own distinct microbiome. It's very different to the microbiome that's in animals other than humans, and very different to the microbiome that's in plants. So if we want to increase microbial diversity, it's not actually going to come from the soil, and it's not going to come from adding something to the soil. There is another source, and I'm going to give you the answer to that question because we've got a lot of slides to get through. So where these microbes are going to come from that are going to give us microbial diversity are actually going to come from seeds. And there's a very good reason for that. And once you understand that they come from seeds, it'll be all very clear. So let's actually start with a seed. What is in a seed and why is it important? Well, we know that a seed has DNA in it. We know that if we plant a tomato seed, we'll get a tomato plant. We'd be very surprised if it grew into a bean. And we also know that most seeds have energy in them in the form of oils and starches and things. We grow cereal crops for their starch content. We grow, sunflowers and canola for their oil. So we're very well aware of the energy component of seeds, and seeds have energy in them so that when a little seedling germinates, it can get going before it starts to photosynthesize. But what we have overlooked until recent times is that seeds actually have microbial DNA. In other words, they bring their own microbiome with them. They bring all of the microbes that they're going to need for their entire life. And if you think of something like let's just pick on a Californian redwood tree or something that can live for thousands of years, all the microbes that are in that plant for its entire life came in the seed. And if you want to reflect on that a little more, think about our microbiome. Where did our microbiome come from? We inherited our microbiome from our mothers when we were born. So we get our DNA from our parents in the same way as a plant gets its DNA from its parents. We get our energy and nutrients, obviously, from our mother as well when we're born, but we inherit our mother's gut microbiome. That's where our gut microbiome comes from. So a seed is basically the microbiome for the next generation of plants. So the plant puts its microbiome into its seed, And then when that seed germinates, that's where the microbes come from. So the microbial DNA forms the plant's core microbiome, and it's encapsulated in the seed. It contains thousands, millions, billions, in fact, of bacteria, archaea, fungi, and protists, all encapsulated in one tiny little seed. So just to give you an example of that, I'm sure you're all aware of what a barley seed would look like. I don't have to describe what a barley seed looks like. You know, think of wheat or oats or barley triticale, they all look pretty similar. There is 9 billion microbes in one single barley seed. Now, on the planet at the moment, we have something like about 8 billion humans. So there are more microbes in one little seed than there are humans on the planet. And you'd think, wow, that's a really microbially rich environment. But in fact, it turns out to be not so rich. There are other environments that are far more microbially rich than a seed. It's just that microbes are incredibly small. It's probably why they have to work together in teams and why we need a large abundance of them to achieve anything. If we look in a much more microbially enriched environment than a seed, like for example, in the rumen of a cow or a sheep, one single drop of rumen fluid, which would be about the same size as a barley seed, contains 1,000 billion microbes. And I can't imagine what 1,000 billion looks like. I can't imagine what 9 billion looks like. So the point I am making is that there are a lot of microbes in a whole range of environments that we can't see. And so we've tended to ignore them, but they are the unseen majority, and they are incredibly powerful. They control a lot of biochemical reactions that take place in our environment. So what do the microbes in seeds actually do? Why is a seed coming along with all those microbes in it? That's a little bit fuzzy, sorry, because it's a blown up slide from another slide I'm going to show you in a moment. But what I want to show you in that particular one is that we're just looking at the red, the yellow, and the green, just representing different microbial groups in that particular bean seed. And we want to see what happens to those in the life cycle of the plant. Well, when the seed begins to germinate, these microbes actually move out into the soil. And they're like scouts on a reconnaissance mission. They send all kinds of signals back to the seed, because the seed can't see anything, it can't hear anything, it can't talk. So the way that seeds communicate with microbes is through biochemical signals, and the way microbes communicate with seeds is through biochemical signals, and the way microbes communicate with each other also through biochemical signals. So they're going to emerge from the seed out into the soil and send messages back to the seed about where we are. Are we, on top of the ground, under a tree, out in the open? Are we on fertile ground, rocky soil? Is it wet? Is it dry? Is it hot? Is it cold? All kinds of information that the microbes actually send back to the seed. And most particularly, they'll tell the seed about the microbial environment that it finds itself in, because that's interesting to the microbes, and it's also interesting to the seed. Now, there's been some very interesting studies done on microbial interactions with plants, and I'm sure some of you would have seen James White's videos or heard some of James White's presentations on rhizophagy, he calls it. Was discovered in Australia 10 years before James White started talking about it, and we called it rhizophagy, but he calls it rhizophagy, so we'll go with that. And he talks about how the roots of plants actually internalise seeds from the rhizosphere and strip them of their cell walls, consume all the nutrients, and then spit them out again through the root hairs, feed them until they're all plumped up again with nutrients, and then internalise them again. So that's the rhizophagy cycle. But the point that I wanted to make about James White is that in one of his videos, or several of his videos, he talks about if you take a seed and surface sterilise it, in other words, you remove just the microbes from the outside of it, not even the ones that are in the inside, the seed loses its geotropic response. In other words, it does not know which way is up and which way is down, because it no longer responds to gravity. So that seed will germinate with maybe roots, sticking up and leaves going down, or roots sticking out to the side, whatever. It doesn't know about putting roots down and shoots up. So that's its geotropic response. That's just one thing that microbes control in a seed. Then we know that they control a whole lot of other functions as well. So they're very, very important to the health of that seed and how that seed responds. So, have we landed on rocky ground or fertile soil? Is just one of the questions that the plant will receive information about from its microbiome. So the plant will then form what we call a rhizosphere microbiome from its own core microbiome. I'm sure those of you who are studying Regen Ag know what a rhizosphere is, but the rhizosphere is basically the area around a plant root, which is a plant root is a rhizome. So the rhizosphere is the area just around a plant root. And all of the microbes that are in that area around a plant root came from the seed. This is where our knowledge now is very different to what we used to think. Like say 10 years ago, we thought that the plant would selectively promote and stimulate microbes from the soil that it wanted to form a relationship with, because we knew the rhizosphere microbiome was very different to the bulk soil microbiome, but we didn't know why. Now we know it's because the plant actually brought that with it. So here you can see, I'm not sure, Simone, is this pointer actually, does this work? Can you see this on the, yep, okay. So inside this bean seed here, we've got some, just shown us some blue microbes and some green ones, just to differentiate them from the soil. Remember, there's going to be 9 billion here. And they're usually just inside the seed coat like that. That's usually where the microbes are located in a seed. And we've got this soil surface here, this brown line. So we've got above ground and below ground. So we've got our little seed is just below the soil surface. First thing a seed always does is produce a root, because it needs to get moisture in order to be able to photosynthesize. H2O is an essential component of glucose that it's going to make during photosynthesis, so it has to get water before anything else. And all of the microbes that are in this plant root, you'll notice that they're the blue ones and the green ones, so they've come from the seed. So as that radical or that first root grows, and then here's a little blue one that's popped out into the soil. So when the plant has developed a little bit more, it's formed this microbiome around its roots. That you'll notice these are the green ones, and the blue ones that came in the seed have actually formed this entire area around the plant root. And then as it starts to produce a shoot, you'll see there's also the blue ones and the green ones are in the shoot. So everything that's living in that plant for its entire life, and it has far more microbes in it, far more microbial cells than plant cells in it for its entire life, it brought with it. And that's a very important point, that the plant brings its own microbiome with it. So this photograph here from Phil Lee is one of my favourite photographs. This is a little root tip here, and it's under high magnification. You can see the root hairs sticking out of the root, and that's where, in the rhizophagy cycle, that's where microbes will actually be spat out and then reconsumed. But I'm not talking about rhizophagy in this seminar. I'm talking about the microbiome that is came with the plant. So you can see all these droplets of liquid here. These are root exudates. These root exudates are designed to feed the microbes that the plant brought with it. So they are very, very rapidly multiplying in the rhizosphere. It is also attracting some microbes that do live in the soil that will live in association with that plant root. And here you can see some droplets of root exudates being cradled. They look a bit like those decoration that people put on Christmas trees, sort of. So they're being cradled by the hyphae of fungi, and these fungi could be mycorrhizal fungi, they could be trichoderma, they could be just opportunistic saprotrophic fungi living in the soil that form a relationship with the plant and form, in fact, very important relationships with the plant. So that's what root exudates are all about feeding the plant's own microbiome and also attracting some beneficial microbes from the soil, but most of the microbes in this rhizosphere, the plant will have brought with it. And how can we tell that a plant is actually supporting its rhizosphere really well? Well, it will have all this soil sticking to the roots like this. So this is a little wheat seedling. It's only got about 2 leaves. It's a very, very young wheat seedling, but it has formed an incredible rhizosphere microbiome very early in its life. So there is the seed there. So these were the first roots that came down from the seed, very rapidly branched, and look how deep they've got. Like, just for a young plant, that is an incredibly deep root system, highly branched with lots and lots of soil sticking from the roots. And then this is the soil level up here, just here. So these roots that came from the crown also have good rhizosheaths on them. A rhizosheath is a column of soil sticking around a plant root. It's a different thing. The rhizosphere is just the area around a plant root. So this is a rhizosphere here too. It goes from the seed up to the crown, doesn't have any soil on it. It never does because there's no exudates come from that. But The rhizosphere and the rhizosheath are not necessarily the same thing. So when we see this good rhizosheath development, we know the plant is producing lots of exudates, and that's a really good sign that it means it's got lots of microbes in the rhizosphere. That's a very critical stage in plant development. If we put high analysis nitrogen, like something like urea or MAP or DAP, or phosphorus like super or starter fertilizer that's got phosphorus in it, or fungicide, which is routinely applied to a lot of seeds, or insecticide, which again is routinely applied to a lot of seeds. If we put those poisons anywhere near an emerging seed, will actually inhibit the formation of an effective rhizosphere microbiome. And that's when we will see seeds like crop plants. You go and dig wheat plants up when they're just emerging, for example, and you'll see those clean white roots if they've been sown with fertilizer or any kind of side. And it is actually very detrimental to the entire life of that plant. So just to go back to talking about the microbiome of the plant, it starts with a rhizosphere microbiome, and then it will form a phyllosphere microbiome. So a phyllodes is a leaf. We talked about a rhizome being a root, and we have the rhizosphere. A phyllodes is a leaf. The phyllosphere microbiome is the leaves and the stems and everything else that's above ground. We just generally use that term for everything that's above ground. So the plant is now going to form its own microbiome, or a phyllosphere microbiome, above ground. So here in this diagram, this is actually from a paper called Microbiome Selection, and it's talking about next generation plant breeding, because the microbiome in the seed is going to be really important for our next generation of plants. And so here on the, well, it's on my right-hand side. I guess it's on your right-hand side too. And we have it, sorry, a rice plant, which is in the grass family. You'll see it has this green microbiome underneath it. Well, they're the microbes that came with that seed. And over here on the left, we have a bean plant, which is in the Fabaceae family, or the legume family. And you see it's got a purplish blue microbiome that came with the seed. So these two microbiomes are very different. And the microbes that are here in the soil between the two is the soil microbiome. These were the microbes that were in the soil to start with, and they're always there. And we can stimulate some of those, but as a general rule, as I said, the plant brings its own. So we also see that there's microbes, all these little dots, all up through the leaves, the flowers, the fruits, everything. And that is a continuation of what started off in the seed, which is now developed into the entire plant. And again, this is very important for the plant. And eventually the microbes that are in the phyllosphere, in other words, the leaves, the flowers, the fruits, they're going to be encapsulated in the next generation of seeds, which is called vertical transmission. And if you want to find out more about this, by reading about it, you'll find vertical transmission in seeds will give you a lot of information. So if we go back to this as the diagram that I showed initially of these little bean seeds with the different colours in them. So we have this complete life cycle where they germinate, grow, end up with those microbes in the leaves, and then form seeds and go back around the whole cycle again. But if we put poisons on around the roots of that plant, then we are actually going to reduce the number of microbes that are in the rhizosphere, and then we will end up with less microbes in the seed. And over time, we find that we end up with dysfunctional plants because their microbiome has been reduced. This plant here has some extra blue microbes shown around the roots and also in the leaves. And this plant will be far more drought tolerant, far more tolerant of waterlogging, far more tolerant to pests and diseases, will grow better, be more productive, and basically fit for life compared to this one with a reduced microbiome. So what we need to know is how do we actually improve the microbiome of our plants, because that's what's going to be the basis of production for our agricultural systems. And the way we manage the soil environment is actually going to determine whether the coarse microbiome is enriched or depleted over time, because it can take some things up from the soil if it needs them. And what we know from the research, and I'm not going to give you examples of this because we haven't got much time today, but there is a lot of research around this to show that the use of high analysis fertilizers and toxic seed dressings, particularly things like neonicotinoids, actually deplete the coarse microbiome over successive generations. So what will happen, just to give you an example, like Rothamsted, which has been running long-term field trials since 1943, so that's 180 years now, and comparing nitrogen, phosphorus, potassium, calcium, like a whole lot of different fertilizers and a whole lot of mixes of those fertilizers. And in recent times, they've done some interesting research where they've shown that by even just using something like nitrogen or phosphorus near a seed, that you reduce the beneficial microbes in the rhizosphere from about 91%. So in other words, if you look at all the microbes in the rhizosphere, they constitute about 91% of them are beneficial to the plant, down to 19%. So, you're basically just knocking all your good guys out simply by putting something like nitrogen or phosphorus near a seed. So be very aware of what you're doing, and then that depleted microbiome is then passed on to the next generation of plants. But there are management practices that enhance microbial abundance and diversity, and they enrich the plant's microbiome over successive generations. So farmers now looking at buying seed for their cover crops or for their pastures or for their multi-species cash crops, whatever it is, are going to be insisting on bare seed because they don't want poisons on the seed. But they're also going to want to know, how is that seed grown? Was that grown in a multi-species crop? Was it grown with biostimulants? Was it grown in a way that enhanced microbial abundance? Because if it was, then I want to buy that seed. So it's important to know where seed comes from and how they were grown, and you could perhaps consider keeping your own, which a lot of people are doing these days. So we have to, just the point of that, seeds carry not only the DNA for the next generation, but also the microbes that will live in association with that plant for its entire life. And when we plant seeds from a wide range of plant families, this is where the practicality of it comes in. We're introducing a diverse array of microbes to soil. Because remember, the bean and the rice plant had very different microbiomes underneath them. So if we had those things growing together, for example, then we would have a lot more microbial diversity in our soil. So when you think about the seeds that you plant, don't just look at them as, I can see here's a sunflower seed, so that's in the Asteraceae family. Here's a corn seed, which is in the Poaceae family. Here's a mung bean seed, which is in the Fabaceae family. Here's some linseed in the Linaceae family. You have to think about the fact that, okay, if we plant all these seeds together, we're putting all these different plant families together, and now we are going to have something that's going to look very effective in terms of soil function. This is why plant diversity is important, because it underpins microbial diversity. So when we have all these different kinds of plants growing together in a multi-species cover mix, or I've just had some beautiful photos sent to me this morning of the mid-row in a vineyard in the Napa Valley in California, and it was just absolutely full of flowers. They've finally got the hint about, please don't put grasses in your interrows. It was like, just all these really, really, really beautiful flowers, which I'm sure the insects would have appreciated and anyone driving past would have appreciated. But it's also meant they had 7 plant families in there. So all that microbial diversity would be doing great things for their soil. What happens when we've got plants close enough for their roots to mingle and they come from different plant species or they come from different plant families? What effect does that have on nutrient availability, productivity, immunity to pests, diseases, cell building, all these kinds of things? So again, we just got our plant here in the Fabaceae family, plant here in the Poaceae family, very different microbes, very different microbiomes. What effect does it have when they're actually growing in close proximity? Well, what we find is that if the microbiomes are similar, they have a negative feedback effect on nutrient acquisition, resistance to pests and diseases, stress tolerance, plant productivity, and probably a whole lot of other things as well. So I've just been in New Zealand, and those of you who visited New Zealand in recent decades would know that you can drive from Invercargill in the south, basically all the way to Auckland in the north, and all you will see is ryegrass pastures. It's incredibly boring. It looks very green, but it's ryegrass, 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 ryegrass. So they have simplified their production systems to the point where they have one kind of grass and they apply insane amounts of nitrogen to try and get that grass to grow, which is why they now have massive effects of nitrate, just poisoned water that will probably be poisoned for thousands of years because of the nitrates that have now leached through into the groundwater. So when we have something like just ryegrass, it means that every individual ryegrass plant or the microbiome underneath that ryegrass plant is going to have a negative feedback effect on its neighbours because they're all competing. They've got very similar kinds of root architecture. They grow at the same time of the year. They're competing for nutrients and light and water. So the microbiome that's underneath those plants actually sends out messages to the microbiomes of its neighbours that we can pick up using sophisticated technology that basically says, get lost, I don't want you as a neighbour. I would rather just grow here by myself and have all this nutrients and water for myself. And well, I won't go into it any more detail than that because I want to talk about what happens when we have dissimilar microbiomes. In other words, where the plants come from very different plant families, they have a positive feedback effect on all of those same sorts of things. And we can measure that biochemically. We can measure that the microbiome is sending out a signal to the other microbiome saying, yes, we like to have you as a neighbour. So why do plants from very different plant families like to have neighbours that are very different to themselves? Well, they have very different kinds of roots. They're what's the word for it? Obtaining nutrients from very different levels within the soil. If you look at something like a deep taprooted plant compared to something that's got shallow fibrous roots, they can actually benefit from being next to each other. They also benefit in terms of they have different microbial DNA and they can borrow microbes from each other if something like a pest or a disease comes along. And if I had time, I would have shown you an example of that, but I can see I'm going to run out of time. But it's one of the reasons that people are using diverse covers now in horticultural situations because they find they don't have the pest and disease issues in their horticultural crop. So let me just give you a few examples of that. One is from the Jena biodiversity experiment in Germany, which has been running for nearly 20 years now, I think. And they looked at having 1, 2, 4, 8 or 16 different plant species growing together in a 20 by 20 metre block. So if you look at this, like looking down as an aerial shot of their research site, and you can see those squares there. So where you can see like a pink square or a white square, for example, that will be some kind of daisy that happens to be flowering at the moment, and that would be a monoculture of that. And then the green squares would be grasses, and then the mottly ones would be mixes of a whole lot of different plant families. So what they did with their 1, 2, 4, 8 or 16 plant species was they took them from 4 functional groups, and those groups were grasses, legumes, tall herbs and short herbs. So if we just wanted to put that into an Australian context, you could say that a tall herb might be something like a chicory plant or a sunflower plant, and a short herb might be something like plantain, for example. So that a 16 plant species mix wasn't just 16 grasses or 16 legumes. You really have to get those other functional groups in there. And they looked at biomass production and insects and microbial activity, water balance, carbon, nitrogen, phosphorus, and hundreds of other things. They probably had at least 100 PhD students working on this because the number of papers that have come out of it have been incredible. So one of the things they found was that as they went from 1, 2, 4, 8 or 16 plant species, they actually got more biomass in those 20 by 20 metre squares. And that has been also the finding from biodiversity experiments taken in other countries around the world, and it's called over-yielding. So if you just have one area of one kind of plant, like wheat or ryegrass or something like that, then it will yield a certain amount. And if you put other species in there, it will over-yield. You'll actually get more yield. And if you want to Google that over-yielding, you'll find it's a very common feature of biodiverse mixes. Now, the other thing that they looked at was what happened to soil depth and soil structure. And this is probably very important when we're talking about regenerative ag, because I'm saying, you know, my definition is that we're restoring function to the soil. And we really want to know, yes, yield is important and over-yielding is great, but what's actually happening to the soil? And one thing we see where we get diversity in our plant mixes is that we see very rapid improvements in soil structure and soil depth as the number of plants in the mix increased. So they have a great little video that you'll find online. The Jena biodiversity experiment just put that in video. So over here on the left, we've got just 2 plant families. So in the New Zealand example again, and this would also be common in dairy in Australia, like in coastal New South Wales maybe, but definitely Victoria, South Australia, southern part of Western Australia, the 2 species or the 2 plant families that have it be ryegrass and white clover. You will always find shallow soil under that. Go out with a spade, dig a hole, and unless it's black soil, what you'll see is this thin black A horizon, could be a couple of inches, could possibly even be 6 inches of darker soil, and then you'll see lighter coloured soil underneath it. You'll see a very distinct difference between the A and the B horizon. That tells you whenever you see that distinct difference between A and B horizon that your soil is dysfunctional. It's not meant to look like that. It's meant to be a gradation. And in fact, you would want it to be a very deep soil. You want to see that dark colour go down for like at least 50 centimetres. So over here on the right, we have a mix that's got the 4 functional groups in it. And what they found in this experiment was that when they had 4 functional groups, they had much deeper soil, much better aggregated soil, much more biological activity in the soil. And you can see This grass that's shown in the background here behind these white daisies, this is the same grass in the background here, it's much taller. So everything has grown better in a multi-species mix than what it did when it was growing on its own. And I've seen that, I was over in Western Australia a bit earlier this year, and where there were grasses growing outside of a multi-species mix, and then this very same grass growing in the multi-species mix, it was 3 or 4 times the height. It was quite incredible. And the landowners themselves hadn't noticed because they didn't recognise it as the same grass. When I pointed out, well, see that grass that's growing under the fence there? That's the same one as what's in your multi-species mix, but it's 3 or 4 times higher. The leaves are much wider and it hasn't gone to seed yet, whereas the one under the fence has. So it just grew a little bit and went to seed. In the multi-species mix, it behaved very differently. So plants will behave differently to what you expect in a multi-species mix, and you can't predict from looking at a monoculture how they will grow. So this is the kind of soil that we want. We want to have this good crumb structure, and these are called water-stable aggregates. If you take one of those pea-shaped lumps and drop it into distilled water, it will stay as a pea-shaped lump and fall to the bottom. It won't disperse and make the water muddy. So whenever we drop soil into water and the water becomes muddy, we know that we don't have good aggregation. We know there's something terribly wrong actually with our soil. So these water-stable aggregates, they're the fundamental unit of soil function, and diversity gives us water-stable aggregates in a very short amount of time. If you've got sufficient diversity, you'll see these starting to form within 6 months, and people will notice that their soil is darker and more crumbly in structure. So water-stable aggregates actually hold moisture within, which actually provides drought tolerance, because once the moisture gets inside an aggregate, it can't evaporate. Plant roots can still get it, so it's still plant available, but it won't evaporate. And water-stable aggregates improve drainage, which reduces waterlogging. So we've got 2 sides of the coin where water-stable aggregates are really important. Again, this is a bit fuzzy. I'm sorry about this, this diagram here, but this is a water-stable aggregate here where we've got these brown, the brown parts of it are the soil particles. And then we've got these little blue circles, of course, is the moisture. So once the moisture is actually inside a water-stable aggregate, it's protected from evaporation. That is really important. If you've got structureless soil, well, for a start, they can't absorb water. And secondly, if they do, they lose it very quickly. So when we've got good soil structure like this, water will flow down around the aggregates and be stored quite deeply in the soil, again, where it is available for later use by plants, rather than being near the surface where it's going to be evaporated. So in this Yena biodiversity experiment, you'll notice that there's a river in the background here. And 15 years into this experiment, that river flooded. And we're all very familiar with these, this sort of scene of flooding, and my sympathies to people who have been affected by flooding. There's been very, very severe flooding in New Zealand just recently too. But what they found in this experiment when it flooded was that they thought they'd actually lost it. They thought, well, that is the end of our experiment, because the water was about knee deep and it sat there for 3 weeks or something. But these plants that were emergent, like there's a plot of grasses here and something else here that probably looks a bit like dock or something like that, some kind of a broadleaf plant anyway. But these were monocultures, and even though they were out of the water, they were emergent plants. When the water went down, these ones died. Here we've got a plot where we can't really even see what's under there. If I look down into the water, I can see there's some little daisies and other little things underneath the water. So they were completely submerged, but when the water went down, those ones survived. And I was talking about this in New Zealand just last week, and a researcher from Massey University, which those of you who are familiar with New Zealand, Massey is on the North Island, and they were affected by that flooding from Cyclone Gabrielle that unfortunately took a lot of lives and caused massive destruction to a lot of farmland in New Zealand. And their trial plots, their multi-species trials, went underwater, just like in this photo. He said that's just what our research trial looked like. And when the water went down, it was the multi-species that had survived, and they lost all their monoculture plots. So they saw exactly the same as what this photograph is seeing. So in addition to this tolerance to waterlogging, diverse plant communities also have much higher tolerance to drought. And that's because these water-stable aggregates can hold moisture within and provide it to plants. So if you've seen my presentations before, you've probably seen this. I can't help putting it in every time because I just love it. This is from the Chinook Applied Research Association in Alberta, which is a province in Canada. And it's quite inland in Canada, so they have It's really hot in summer, like plus 40 degrees, and really cold in winter, like minus 40 degrees. They have this huge swing in temperatures. But July is the middle of summer, and they basically grow everything in summer. So what we would think of as a winter crop is also grown in summer. So they had a triticale monoculture that they were in the process of planting on the research station, and the local farmers had just been to a seminar given by Gabe Brown, who lives in North Dakota, which is just on the USA side of the border between Canada and the United States. So he's not that far from Alberta. And he had been talking to them about, back in the day, what they call cocktail crops. And the local farmers said, can we please see what a cocktail crop looks like? And the Chinook Applied Research Association said, well, we don't really have any land to be looking at, extra land to look at cocktail crops. But we could put one in over the top of this triticale monoculture that we've just planted, because it's in a large paddock and probably wouldn't hurt to put a little area of a cocktail crop over the top of it. So the trit was planted one day, and the very next day, so there's only a day difference in these, they oversowed it with a crop containing oats, tillage, radish, sunflower, field peas, faba beans, etc., etc. But the main thing is there was 4 plant families there. It was a very hot, dry summer. The triticale failed. It wasn't going through to yield. You can see it's not really a very dense crop, so it wasn't plant density. That was the problem. You can also see, if you've got a good eye for soil, that the surface of the soil is very compacted, looks very ordinary, and we'll find plenty of soil that looks just like that in Australia. So it's not a healthy situation. And in a situation like that in Australia, a farmer would say, okay, we've had a drought year, our crops failed, we've got crop failure, and it's the weather is the problem. Well, actually, no, it's the lack of diversity that's the problem, because here is the cover crop that was sown over the top of the triticale. So it still has the same density of triticale in it, but on top of that, we have sunflowers and radish and faba beans and peas and all those other things, 9 other things planted over the top of the triticale. So you'd think in a drought year, with all those extra plants, wouldn't they run out of water? Well, that's not what we see. So that's the comparison between a monoculture and a polyculture. And we've now got people multi-species cash cropping here in Australia and getting fabulous results and basically making thousands of dollars per hectare more than their neighbours whose crops are failing either for weather-related reasons or pest-related reasons. So down south, Victoria, South Australia, etc., just huge issues with red-legged earth mite that we're not seeing. We're seeing them in monoculture, but not in polyculture. So nutrient acquisition is another important function that we want our soils to perform for us. And again, microbial diversity is absolutely fundamental to nutrient acquisition. So this is from another research station. This is Ontario, which is another province in Canada. Why are we not doing these kinds of trials in Australia? It's a very good question. But here we have a monoculture of radish. And this plot is about 20 metres wide. So they have these like 20-metre wide strips that are probably about 50 metres long. And it was a cover crop demonstration trial. So they had about 12 different cover crops grown as monocultures. And then there's 3-way mixes, 6-way mixes, 9-way mixes, and 12-way mixes, all beautifully labelled. So the farmers in the district could come and walk along the rows and look at, well, this is what a monoculture of radish looks like, for example, or a monoculture of sunflowers. And then this is what happens when I put all these things together in various combinations. And they were able to make decisions about what they may like to have on their own farms as a cover crop. So this cover crop of radish monoculture is looking very nitrogen deficient. So everything in this trial all received the same amount of fertiliser, but when this is grown on its own, it's nitrogen deficient. And just right next to it, the plot over here on the right-hand side, growing side by side with this one, also has radish in it, but it has some oats and some sunflower and some phacelia. So these leaves here are the radish leaves. Same radish, planted on the same day. Everything was the same, exactly the same soil, the same weather. It's 3 times higher, it's much greener, it's really lush and green, and we've got a little sprinkling of, there's a sunflower there that also has very dark green leaves. We've got a few oats that you can see are starting to head up through the crop here, and we've got a phacelia plant here, which is in Boraginaceae. So we've got Boraginaceae, Poaceae, Brassicaceae, Asteraceae, 4 plant families, all you need. There are no legumes in there, and there is no nitrogen deficiency. You don't need legumes for nitrogen, you need diversity for nitrogen. So if I splice that photograph and put it next to the previous one, which shows you the 2 things side by side, that is the difference that a little bit of diversity makes. It's quite extraordinary. So the unlocking and the transportation of nutrients requires a team effort from many different kinds of microbes working cooperatively. And when we have those different plants growing together, their microbes, they all have their own microbiomes with them. Their microbes work together to do what the plants need. And we can't tell from a monoculture how plants will actually perform in a polyculture. So you can't grow one thing on its own and say, oh, this is what will happen when we put it with something else. I'm just going to show you this little experiment that a wine grower or a grape grower in California undertook. He realised that he needed to have some plant diversity in the mid-rows of his vines. He bought 12 different kinds of seed from green cover seeds in the United States, and then he realised that he actually didn't know what these plants looked like. He knew a lot about grapevines. His father was a grape grower, his grandparents were grape growers, whatever. He knew all the different varieties of grapes. He didn't know what barley looked like or chickpeas or faba beans or those kinds of things. So he went to the hardware store and he bought 12 little pots and a bag of potting mix, and he put each of the species in their own pot so he could recognise them when he saw them out in the vineyard. But then he also thought, well, I'm not going to grow them on their own out in the vineyard. I'm going to be putting them together. So he bought another pot, which is about 3 times larger. It wasn't 12 times larger. And he put all the 12 species in the one pot together. And this is what happened. So you can see that the ones that are in a pot by themselves, many of them look incredibly nutrient deficient. So I'm not sure whether you can see this little barley pot of barley plants down here. They look a bit like those radish looked like when they were growing on their own. And if that was a field of wheat or barley or oats or something like that here in Australia, farmer would go out and look at that, or a research scientist would go and look at that and go, you know what, that's nutrient deficient. And they'd be right. And they'd apply some nitrogen to it and it would grow better. And you go, oh, you can't grow a crop without nitrogen. So we form that conclusion because we've looked under a certain set of circumstances that have created that result. When we put that same barley in a mix with all these other things, so we've put all 12 together now, there is nothing in there that's nutrient deficient. And even something like, so these are the faba beans at the back here, they're looking really yellow too. And supposedly, he said all the seed was inoculated, so supposedly they should be fixing their own nitrogen, but look, they're like 3 times bigger when they're here in the mix. So you can't tell from a monoculture what plants will do in polyculture, and we've made all the wrong decisions about fertilizers when we've just about every experiment's ever been undertaken in any research facility in Australia or overseas has been undertaken with monoculture. So anything that, it doesn't matter what aspect of plant growth has been looked at, if the plants were grown in monoculture, you can throw that paper out because it doesn't mean anything. It's not relevant. Nature doesn't grow as a monoculture. Our natural systems didn't function as monocultures, and we have to make sure that all of our systems in agriculture, whether they're cash crops, whether they're horticulture, whether they're pastures, we have to make sure we get plant diversity in there. Otherwise, we're going to be on this input treadmill that not only is going to cost us a lot of money, but it's also going to be very detrimental to our soils, to our waters, and to the oceans where the water flows. So in a monoculture, there's just insufficient diversity of microbes to carry out all the different tasks. It's as simple as that, really. So let's look at frost tolerance. This is another example of resilience to stress that really kind of blows my mind. Whenever I see these sorts of photos, I see them from around Australia. People are always sending me photos of this, and I love it. And these ones are particularly clear. These come from Mount Gambier, from Cam Banks, who was an organic dairy producer in Mount Gambier. So this pasture in the front is ryegrass and white clover. The pasture, so that's 2 plant families, Poaceae and Fabaceae. This pasture in the back is the same cultivar of ryegrass and white clover that was oversown with chicory and plantain. So we've got 4 plant families now, because chicory is in Asteraceae, the daisy family, and plantain is in Plantagenaceae. 4 plant families gives us like 2 or 3 degrees of frost tolerance. Really quite extraordinary what a difference that makes. And here is another photograph where we have ryegrass and white clover in the foreground and ryegrass, clover, chicory, plantain in the background, and a few stinging nettles just for good measure. So here we can fence out a frost, but we can also fence out pests, diseases. We will see, in fact, I've been to this farm and we walked around here with a spade. Soil structure in this paddock was incredibly good compared to this one. it's just, I mean, you have to see it to believe it. And I would like to see many more people experimenting with diversity to see for themselves on their own farms, or if you don't have a farm, in your garden, and just see what happens with plant diversity. So I asked Cam if he would use a refractometer to go and measure the Brix levels of the ryegrass in both of those paddocks. So if we go back to there, what is going on? Like, that's pretty amazing to be able to fence out a frost. There's got to be something going on here. So I hope most of you, in fact, I hope all of you have a refractometer. I hope you use it. I hope you understand what Brix is and why you're measuring it. So Brix is a measure of the dissolved solids in the sap of the plant. If we put distilled water on here and close down on our slide, close down the plate over the top, look through the other end, holding it up to the light, we would see that distilled water has a Brix of 0. In other words, there's nothing in it. If we were to put some sugar in that, we'd find that the Brix level would go up. Depending on how much more sugar we'd put in, that we would get a higher and higher Brix level. Now, if we were to take distilled water and put it in the freezer, it would freeze at 0. If we put some sugar in it and put it in the freezer, it will freeze below 0, because when there's dissolved solids in the water, it freezes at a lower temperature. So we can get 2 or 3 degrees of frost tolerance simply by having a high Brix level in our plants. So Cam went out and measured the Brix level, and just from memory, I think he said it was about 4 where there was ryegrass and clover, and about 8 where the ryegrass was growing in with the chicory and plantain. So doubled the Brix level. I would still like to see it up around 16 at least, but at least it was better than it was in the low diversity plot. So what's good for soils is also good for animal production. I can see that I'm running over time. I don't know how many graziers we actually have in this group, but I'm just very quickly going to show a couple of things about animal production and then open it up to questions, because I did want to say something about animals as well as soils and plants. So Cam, who's sent me those photos of frost tolerance, he also found that milk production increased 4 to 5 litres per head per day on those diverse swards compared to the ryegrass pastures. That's a 20 to 25% increase in milk production. And you will see the same kind of thing with growth rates in lambs. Coal size has got some really good data on that. You've got double the growth rate in lambs on a multi-species pasture compared to a barley pasture. And we also see the same thing, obviously, with beef, that you'll see that your production stock will increase weight much faster. So if we have a look at the impact of forage quality on average daily gain, so these are the Brix levels along the bottom here. I mentioned it's a measure of dissolved solids in the sap. So it starts at 0, but we've got 1, 2, 3, 5, like all the way up to 26% here. And then on this axis, we have average daily gain. This is in pounds per animal unit per day because it's American data. There's almost a straight line response. The more you increase the Brix level, the faster animals will gain weight, or that could also be milk production. You'll see something similar in milk production, the higher the Brix level of the pasture. Now, if you apply something like nitrogen to pasture, Brix immediately falls. Why does the Brix level fall when you apply nitrogen? Because you've taken away the job of the microbes. You've just supplied nitrogen to the plant, doesn't need to produce root exudates anymore, doesn't need to support microbes in the rhizosphere. And when it's not supporting microbes in the rhizosphere, it's not taking up all the minerals and trace elements and everything that it needs from the soil. So we will end up, because Brix is not just sugars, it's also minerals and trace elements, and we will end up with very low Brix in pasture, low production figures in our livestock, and probably all kinds of animal health issues that relate to that lack of trace elements. So again, just to use New Zealand example, I'm going to pick on them because it's just there, just come back from there, but we see ryegrass monocultures Brixing here around 1 to 3%. And we can get them right up. We saw some right up at 17%, where people had good diversity using biostimulants, getting right away from synthetic foods. So your milk production is going to be much, much better. And also we've seen empty rates, in other words, the number of cows that haven't fallen pregnant on a dairy farm in New Zealand this year hit 30%. Can you imagine running a dairy with 30% of your cows not able to conceive? And Dairy New Zealand is having all these big conferences and things and about, oh, is it the weather? Is it this? Is it that? Well, I'll tell you, it's the ryegrass and the nitrogen that is the problem, because they're Brixing down here and the poor cows can't conceive. So we're seeing, they can't conceive on that such poor quality pasture. We're seeing where farmers have moved to diverse pastures, they've got their empty rates down to like 2 to 3%, compared to the national average. I think at the moment is 24%. Unbelievable for dairy. Anyway, animal health. The other thing about diversity is not only good for soil and nutrient acquisition and soil structure, all of those things, but diets that are rich in secondary plant compounds. These are the things that have these big names like bioflavonoids, carotenoids, polyphenols, anthocyanins, all these unpronounceable words. They're important in our diet as well. They're often in the colourful things, like capsicums and carrots and whatever, where we find our secondary plant compounds. They're not in grass, not to any measurable extent anyway. And animals need to have these secondary compounds. We're talking about ruminant animals now, because microbial diversity in their gut is dependent on the plants that they consume, because remember, they're taking in plants that are full of microbes, and the more different kinds of plants they take in, the more microbes they've got in their gut, and that improves their feed conversion efficiency and their immunity. So we see much more conversion of feed to either milk or live weight when we have these diets that are rich in secondary plant compounds. So let diversity do the work. And I've just got one more little thing I wanted to show you. The Irish have actually got this. They've been running a Smart Sward project for about 10 years now. And this is just like one little result that came from this project. With ryegrass with 150 kilos of N per hectare per year produced 9.4 tonnes of dry matter. When they increased the amount of fertiliser, they got more dry matter because ryegrass is very responsive. It's a bit like that barley plant I showed you. It's yellow if you don't feed it. But when they had a 6-species sward, which was ryegrass, timothy, which is another grass, red clover and white clover, 2 legumes, chicory in the daisy family, and plantain in Plantagenaceae, with no fertiliser, they got 12.5 tonnes of dry matter per hectare. So diversity replaced fertiliser. And the Irish government has been battling with high nitrate levels in groundwater, salmon dying in the rivers, tourists not coming anymore because of all the nitrate toxicity, etc. So they have introduced a incentive program to get farmers to stop planting ryegrass basically and start planting diverse species and stop using fertiliser. So the Irish Department of Agriculture, Food and the Marine last year introduced a pilot scheme called the multi-species sward measure, where they paid farmers €50, or they contributed €50 towards the cost of a 12-kilo bag of seed. Ireland, the Republic of Ireland, is in the European Union. They're not part of Brexit, and they still use euros as their currency. A 12-kilo bag of ryegrass seed costs about €100. But if you bought a 12-kilo bag of multi-species seed instead, you received an incentive of €50. In other words, you got it for half the price. And that worked really, really well, encouraging farmers to plant multi-species and not use fertiliser. This year, their multi-species sward measure has been expanded, and farmers are paid €300 per hectare. Oh, sorry. In New Zealand, that's $540 per hectare. In Australian currency, that's about $500 per hectare to plant a multi-species mix. Can you imagine that the Australian government actually paying farmers $500 a hectare to put in multi-species in place of single species? But the farmers must plant the species that the government says, which is, I guess that's typical of a bureaucratic scheme, that it must be these particular species. So this is on their website. You have to plant perennial ryegrass, timothy, white clover, red clover, plantain and chicory, and they specify the weight and the number of kilos per hectare. So I guess if the government's going to pay you that much money, they're going to have a say in what you plant. But it's very interesting because we've got 4 plant families here, and these are the plants that have come up time and time again in a whole lot of different environments. Now, obviously in Australia, that would be different. We wouldn't be looking at planting something like timothy, for example, but there are plenty of other, you could put, it's just gone straight out of my head. Americans call it orchard grass. Anyway, can't think of the name. Anyway, there are other things that we could plant that would be much more suited to our environment. And the multi-species sward measure, there's been all these press releases and everything in their newspapers from the government saying that if you plant these multi-species, there's a reduced need for fertilisers, there's reduced input costs, there's improved soil structure, there's improved drought tolerance, improved water quality, enhanced biodiversity, and they are basing that press release on all of the studies that they have been doing in Ireland for at least 10 years, looking at what biodiversity actually does for your soil. So it would be lovely to see at some time in the future that we had some press releases coming from our government agencies saying that their results have been showing there's similar things, but my understanding is that any of the research that's been undertaken by government agencies to date has actually had fertiliser put under the seed, which is the worst place you could possibly ever put fertiliser. So they're not going to see any benefit from having multi-species. You basically nuke your whole experiment if you start putting high analysis fertilisers near those seeds, for all the reasons that I mentioned earlier about seeds and why they're important. So we've tried to simplify agricultural systems to make them easier to manage, when what we really needed to do was to make them more complex. And fertilisers are often viewed as a necessity when they are a substitute for biological diversity. And one more little thing from Ireland, sorry I have gone over time, but we started a bit late. But these green bars here are perennial ryegrass at different rates of nitrogen fertilisers. So we've got 0, 120, 240, 360. That's 360 kilos per hectare. So if that was urea, it'd be like, 600 or 700 kilos or something of urea per hectare. And ryegrass really responds to that, which is why with monocultures, and also even with cash crops like wheat and that, I can see why people put fertiliser on, because you get this response. But over here on the right-hand side, this is the mix which the government is now paying farmers to, actually basically paying farmers to plant. We've got the perennial ryegrass, timothy, red clover, etc. And here with no nitrogen on it, so we've got again none, 120, 240, 360 units of N per hectare, with no nitrogen on that diverse mix. It's basically as good as the ryegrass with all this. There was no statistical difference between, say, for example, 240 units of N per hectare and a perennial mix or a multi-species mix with no nitrogen. So this was one of the reasons that the Irish government said, we need to incentivise farmers to plant diverse mixes and get away from the fertilisers. So the drivers for soil health are going to be plant diversity, a minimum of 4 plant families, not species, 4 plant families, and use of biostimulants in place of high analysis fertilisers. So I'm just going to, if people actually are planting pastures, I know not everyone is, but people will be planting cover crops, I hope, will be planting cash crops, we'll be putting multi-species mixes in horticultural situations. So just please use a diverse mix, use a biological fertiliser in place of inorganic N and P. And in a pasture situation, it's actually going to do all of these things for you: improve animal nutrition and growth rates, milk production, conception rates, reduce your dependence on vets. But I don't know how many graziers we have in the room or how many crop producers, but I'll turn that over to questions now. I'm sorry that we don't really have much time, and I didn't give you an opportunity. I didn't sort of talk about some case studies of where there's been dramatic reductions in pests and diseases, etc. But open to questions now. So Simone, would you like to?
Certainly, Christine, and just huge thank you. That was an amazing, amazing presentation. I don't know about others, but I don't mind it went over, because I think what's really valuable is your ability to share all of these findings with us. I know I'm delving into scientific reports and studies and publications isn't for everyone, and to see it presented so clearly and factually was just fantastic. So thank you so much. There are a few questions, so I'll just share those with you. So Lorraine Gordon has said, thanks, Christine, awesome presentation. Should farmers inoculate their seeds or not?
If you're talking about legumes, yes, they will still require their Rhizobium inoculant, because Rhizobia don't come with the seed. It's one of the few microbes that the plant actually doesn't bring with them. Legumes will form a relationship with Rhizobium in the soil if they're there. So if that plant's been in that soil before, they will be there. But if it's a new crop that hasn't been planted in that soil before, you do need to inoculate them, yes.
Did you want to keep going, Lorraine? Did you want to put your mic on?
Oh, hi, Christine. Yeah, Lorraine Gordon, great job and fascinating. And blimey, you can talk fast to get through a lot of material. Well done. The only other question I had was about introducing foreign microbes into a landscape that already has a pretty good microbial population. Should we just be sort of feeding them some fish supplements or seaweed supplements to feed our microbes, or really not do anything at all?
So we've introduced foreign people, foreign plants, foreign animals, foreign earthworms, just about everything is foreign as far as our soils are concerned. And the microbes that are in the soil are not as a general rule, plant-associated microbes. Their microbes just carry on their own little, live in their own little worlds in the soil. If we want to have flourishing plants, we do have to bring plant-associated microbes. So you're not going to find those microbes in something like compost or fish or any of the other things that you mentioned. They will not have the plant-associated microbes in them, but we can certainly help the plant to support the microbes that it brings with it by applying a biostimulant. So I'm very much in favour of farmers using biostimulant. As the word suggests, it stimulates biology, and we want it to stimulate the biology that the plant brought. So the plant will bring its microbes, it then has to feed those. So every microbe that's in a plant has to get its energy from somewhere, and the plant's feeding it, which means it has to photosynthesize faster, which is one reason why its Brix level is higher. And during that process of photosynthesis, it's channeling a whole lot of energy down to its roots and producing those root exudates to feed the microbes that it brought with it. But if we use a biostimulant on the seed or on the leaves, we can actually help the plant undertake that process. If we use something that's a replacement for that process, like nitrogen or phosphorus, for example, which actually replaces a natural function that those microbes would have performed, then we reduce photosynthetic rate, we reduce Brix levels, we reduce animal nutrition, we reduce soil structure. We actually get in the way of, like I said right at the beginning, it's a process-driven, we actually want process-driven agricultural production. We have to understand what those processes are. So we have introduced plants from overseas and animals from overseas. Those plants will bring microbes with them, and there's nothing wrong with them being from overseas as well.
Amazing. Thanks, Christina. I'll come to you in just a minute, Peter. I've just got Claire and Craig have just asked if we were planting a mix and we could only find a coated seed for diversity, do we put it in or leave it out?
Oh, that's a really tough question. I think that if you look around, you will find in Australia, you will find now people with bare seed for everything, because there's so much demand for bare seed that I would leave it out. And in fact, I would give the seed supplier a blast and send it back and tell them. So, I mean, one of the common coatings that's used on seeds in Australia, it'll come under the name of something like gaucho or poncho or something like that. Sounds quite innocuous, but then you have to look that up on the internet, find out what the active ingredient in that is. And gaucho, which is on a lot of pasture seeds, I don't know about crop seeds, but probably is also gaucho, is a neonicotinoid. That is one of the most potent poisons that have ever been released into the environment. About 2% is taken up by the plants. The other 98% gets out into the soil, out into the waterways, kills freshwater invertebrates. Like, you can forget about, you know, your frogs and all that. And like this photograph here of the bees, it kills bees. It's been one of the main poisons that's been blamed for bee colony collapse is neonicotinoids. It has been banned in just about every country in the world. Australia and New Zealand are about the only two countries that are still using them. So please don't put those poisons, and please don't put those poisons on seeds, and find out what they are. If you get seeds and it comes with all this coloured stuff on it, go back to the seed supplier, ask them what that is and what those ingredients are, and look it up yourself on the internet. Find out, you know, and you'll need to look at overseas data, because unfortunately the Australian, you know, veterinary pest, whatever people they're called, that tell us whether these things are all right or not, will tell you it's perfectly safe, you know, just kills all your frogs and bees and stuff, don't worry about it. It's not safe, and they're not safe, and we shouldn't be using them, and we have to send a very clear message to seed suppliers that we will not buy seed with those on them. So you need to get that seed from somewhere else, and you need to tell the seed supplier that you don't want it on there. They put it on there. The seed supplier is the person who put it on there. Tell them not to do it.
Thanks, Christine. Peter, did you want to take your mic? I can take your microphone off if you just want to. Yeah.
Can you hear me now?
Yeah, that's great.
Yeah, my question, it's actually a horticultural question. I'm growing blueberries, and I'm growing them with 15 different species of native plants alongside and between the blueberries. There'd be 4 different plant families in there, and there's grasses, legumes, shrubs and trees. My question relates to ericoid mycorrhizal association. So I've found this plant growing down by the tractor shed, which is a member of the Ericaceae family called Tropicarpa. And there's not too many Ericaceae in the subtropical area where I'm living. And I'm wondering what's the best way to get the ericoid mycorrhizals happening within the blueberries where I'm growing them. I've actually soaked them with a commercial brand, like the blueberry plants, I soak them in an ericoid mix. But now I'm thinking, if you're saying the mycorrhizals go through the plant, and if I literally take cuttings of that Tropicarpa, will I be getting any influence of those mycorrhizals in that plant if I put them into the patch? Okay. What's the best way to get the mycorrhizals in, basically?
It's interesting, Peter, because the two questions I've been asked, one was about nitrogen fixes in legumes, which don't come in the seed, and the other one is mycorrhiza, which also don't come in the seed. So they're the two very well-known examples of where the plant actually forms an association with microbes that are in the soil. So your mycorrhiza do have spores in the soil, not in the plant. And the cuttings won't have mycorrhiza in them. They won't be in the stems. The spores will be in the soil around the roots, and it would be a lot of trouble to try and get that ericoid growing with every single, right next to every single blueberry plant that you've got. But one thing you can do is try and multiply that ericoid up in pots or something like that, and take a little bit of the soil from those pots and put it around your blueberry roots. So that the soil around the roots of that plant that you've found that will have those ericoid mycorrhiza in it, all the soil around those roots will also have that mycorrhiza and the spores of that mycorrhiza in it. You can go to a lot of trouble. You can what they call like wet wash the soil, and you can wash the spores out and everything. But I would suggest that if you could maybe just take a little bit of that soil with your blueberries, maybe when you first plant them, if you were planting anymore, or even just putting, just like a tiny amount, like a teaspoon or something, just like maybe dig a little hole and just so it's in the ground near some roots, and but wouldn't they already be inoculated with ericoid mycorrhiza? Well,
that's a good question. I don't know, because where did I get them? I got them from dailies. They might have been, but there's no guarantee that they were. And I was just, in terms of a practice of getting those mycorrhizals, I was just interested in if, also the other thing is, how does it, like nature does this, if once you start planting out an area, what I've found is I'm already getting native species coming up, that are, self-sowing, like red ash, and there's a whole lot of things coming up now that I've sort of cleared the area and put other plants in. And I'm just curious as to how the mycorrhizals just show up, like, they're in the soil, I get that, but they must be able to spread over quite vast distances so once you increase your forest area and you start linking up areas of forest to, say, the blueberries, do you get this movement of mycorrhizals over distance? Or is it fairly contained within a given area?
I wouldn't know enough about ericoid mycorrhiza. I can tell you that ectomycorrhiza, which are the ones that will be forming relationships with your trees, do move over very large areas. Like in forests, all the trees in a forest will be connected with an ectomycorrhizal network. And presumably it's possible that blueberries can also link into that. I'm not sure how specific the ericoid mycorrhiza is. I don't know enough about it. I do know that the endomycorrhiza that we find in grasses and legumes will link up with ectomycorrhiza in trees. So we know they will link up, but whether or not ericoid are just like even Orchidaceae, like Orchidaceae family have their own specific microbes as well. And some of those are generalists and some of those are very specific. So there'll be certain, like, little terrestrial orchids, like not in green hoods and things like that. Some of those will have very, very specific mycorrhizal associations, and some will be generalists and then just link up with any. So I don't know where blueberries form on that spectrum, whether they can link up with other kinds of mycorrhiza or specifically have to be ericoid. Don't know enough about that, sorry.
Yeah, that's really fascinating. Really enjoyable, and so much knowledge that I'm just really excited about, the information that's coming out. So it's
Yeah,
really
I
great
mean,
to hear your talk.
when there's so much stuff on mycorrhiza, like it's a fascinating, I can spend hours reading about mycorrhiza, but I don't usually read about the ericoid ones because I don't grow blueberries. But I think that the deeper that you go into that, the more you'll find maybe information about how to propagate the spores of the ericoid so that you can get them more general.
Thanks, Christine.
Well done for your diversity. Like great that you've got diversity in your blueberries.
Yeah, it's making a difference already. Yeah.
Because most of the ones that come out of Peru, for example, are hydroponic. There's organic blueberries just flooding the market from other countries, and they're all hydroponically grown.
Yeah, I find that here, like where I'm near the Coffs Coast, and it's pretty tragic. Like it's monocropping, and you're talking about those neonicotinoids. There's a lot of pesticide and there's a lot of chemical use that is basically going straight into the, it's ending, it's right on the coast. So it's heading into the oceans, it's ending into our rivers. It's pretty tragic. So.
And in our food chain. So if you measure look at pesticide levels in our children, they're not something to be proud of. We got very, very high levels of pesticides in our children here in Australia compared to other countries.
Yeah.
Then they have blueberries for breakfast, like that, not yours, but someone else's loaded with pesticides.
Yeah, well, they're promoting blueberries as, this wonder food, this antioxidant, it's a wonderful, but you look at how they're grown and it's pretty tragic. I don't think it's going to be a healthy product for you. Even the organic ones, as you say, are largely hydroponic.
Yeah, exactly.
Thanks, Peter. Thank you for your contribution. That's fantastic. We do have another question coming from, is this correct, Gabriella and Michael, who have asked, is mushroom compost good for everything, or are there some places better not used?
I'm not sure what you mean by better for everything, but again, we're talking about, when you're talking about any kind of compost, you'll have microbes that are in that compost that are, like involved in the breaking down of organic matter into smaller particles, etc. Those kinds of microbes don't survive in the soil once the compost has all been used up. So they're very beneficial. While they're there, compost is very beneficial, a very beneficial addition to soil, but you're not actually introducing microbial diversity. You still have to have all your different kinds of plants, and you can support them with mushroom compost. Again, as Peter was saying, with blueberries, we think of blueberries as being healthy because of the antioxidants and the secondary plant compounds that they have in them. But the way they're grown is often very unhealthy because there's massive pesticide use, and often those pesticides are applied close to harvest and they're often off-label. They haven't actually been even allowed. They're not even registered for use on blueberries. That's not to say all blueberries are bad, but similarly with mushrooms, surprisingly, although mushrooms are a fungus, a lot of mushrooms are grown with high fungicide use to try and stop the other kinds of fungi from basically contaminating the mushroom. So we think of them as clean, but it depends how they're grown. So and that's why mushroom compost is often regarded as being a contaminant, because it can be very high in fungicide. So again, it just depends how these things are grown. You have to find out more about the product, really.
Did you want to add to that?
Thank you.
No, that was helpful. Thanks. And again, I guess it's coming from a monocrop, so it's limited in what it's introducing.
Look, most composts are good. I wouldn't specifically be able to say something about mushroom compost, but I know I have a big question mark about mushrooms. If I'm going to buy mushrooms, I want to know how they were produced, and I would only buy organic ones because I know that fungicides are used in mushroom production, and that I think mushroom compost is often fumigated or sterilised or something, isn't it, before it's sold? I don't know enough about it. Sorry, you'd have to look more into it yourself. I can't answer that question. I really only know about the microbes that grow in association with living plants, and I know that those microbes, that we need a diversity of plants to support a diversity of microbes to really get our soil to function. And as a general rule, compost is beneficial, but you have to look at how that compost was produced, and if it's in an environment where fungicides and insecticides and other nasty things are being used, then it's not going to be good.
Thanks,
Thanks.
Christine.
Thank you.
Yeah. Well, there are no more questions have come up, or no more hands have popped up either, Christine. So Peter's got one more. Quick one, Peter. We're nearly at time.
Just on soft rock phosphate. NTS comes out with this, like soft rock phosphate. It's, I'm just wondering what your thoughts are on, applying.
So as a general rule, Peter, you will have quite a high phosphorus level in your soil, because Australian soils are very old and deeply weathered, and that's why phosphorus doesn't show up even though it's there. Because when we do a test of available P, like Bray P, Morgan P, Colwell P, something like that, one of those tests will actually just show you what's in solution, which is orthophosphate, which plants can take up straight away. It doesn't tell you about how much total phosphorus you have. So one thing you'd need to do before spending money on something like soft rock phosphate is actually ask for a total phosphorus level. Like, so you could send a sample to EAL, for example. I don't know who you use as your lab. Yeah. Just ask for total P. Usually they do total P now, because I used to hammer them about that. So you've got to tell people, but maybe you still have to specifically ask for, and so you'll see they give you Bray, Morgan, Colwell, etc. But you need to ask for total. And the other thing is, it's very important to do a leaf test on your plants and find out how much phosphorus they've got in them before you start worrying about applying it, because you might find that a soil test is showing your available phosphorus is quite low, but in the leaf you might find it's perfectly fine. And what that means is that your plants have actually formed a relationship with Ericoid mycorrhiza, or even tapped into some of the other kinds of mycorrhiza, which are very important for getting phosphorus. Your mycorrhiza are the most important things, the most important microbes for getting phosphorus for your plants. That's their key function, is to provide phosphatase enzyme, or support bacteria that produce phosphatase enzyme, liberate that phosphorus that's locked up in your soils and transport it to the plant. So your mycorrhiza are the most important thing for your phosphorus, not how much phosphorus you apply. Now, the thing about applying soft rock phosphate is it's not in a readily plant available form. So what you're doing is actually just adding it to the already pool of non-available phosphorus that you have. So you've just put a few more drops into the ocean, basically. It sounds like a great, it's a bit of a marketing gimmick, really, I think, soft rock phosphate, because I say, well, it's not immediately plant available, so it won't interfere with mycorrhizal associations, etc. But it also is not immediately plant available, full stop. And you've already probably got a lot of phosphorus that's not immediately plant available.
Right.
And you need to find out through EAL, ask them for total phosphorus, and do a plant tissue test through them, if you like.
The plant tissue test, your bricks doesn't show up phosphorus, does it? That just shows up your sugars.
Brick shows up a lot of different things, including phosphorus. So it will show you minerals. When you get a really fuzzy line on bricks, it means that you've got a lot of minerals and trace elements. So if you get that really straight line, it means that just tells you what the sugar level is. But if you get a very fuzzy line, sometimes the line's so fuzzy you can't really even see where it is, exactly where it is, that will tell you that you've got lots of minerals and trace elements. Now, you don't know what those minerals and trace elements are, unfortunately, and whether they include phosphorus, but the chances are, if you've got a really fuzzy line, that phosphorus will definitely be in there, because plants need phosphorus. They can't grow without it. And that's why we put so much emphasis on phosphorus, is because plants won't grow without it. But you need your mycorrhiza to get it into your plants. And your mycorrhiza are definitely stimulated by plant diversity. So it sounds to me like the things that you've done, like having all those different kinds of plants there, is the key. And if your tissue test or your leaf test is showing that phosphorus is in the right area, then you don't have to worry.
Yep.
Nutritech have some good products, but they will also sell you things you don't need, as anybody will, because they'll make money from it. So reactive rock phosphate it's an interesting one. You're just actually adding more non-available phosphorus to your soil. That's what you're doing. So find out how much you've got to start with.
Yep, great. Thank you.
Thanks, Christine. We do have one more from Gabriella and Michael as well.
Yeah, Christine, just wanting your comment on things like the manufactured BAM product from NTS, beneficial anaerobic microbes. And are there some aerobic microbes that we want to add? I know the difference between aerobic and anaerobic as a concept, but how's that relating to the soil per se?
Okay, so the soil has anaerobic and aerobic microsites in it, and the anaerobic ones are probably the most important ones. And the rhizosphere itself will be relatively anaerobic if you have good rhizosheaths around the roots, that column of soil around the root. The inside of a rhizosheath will be relatively anaerobic, and it needs to be anaerobic, not totally, but relatively speaking, because the microbes that use nitrogenase, which is an enzyme that microbes use to fix atmospheric nitrogen, is inhibited by oxygen. So nitrogen fixing can only take place in a region either around plant roots or in nodules on legumes that have a low partial pressure of oxygen. And people think that anaerobic is bad, but in fact, our gut is anaerobic. And if someone injected a whole lot of oxygen into your bloodstream, you would also probably die. You know, like, I mean, you don't want to have oxygen in your bloodstream. You don't want to have, added oxygen. I mean, I know our blood is carrying oxygen, but, you know, like your gut is anaerobic. The rumen of a cow is anaerobic. Like this anaerobic fermentation process that takes place in the gut of earthworms, which is why vermicast and vermiliquid and those products are so valuable for soil. So we have to have both aerobic and anaerobic processes taking place in all living systems. BAM is a good product. It is an expensive product, but it is a good product. You could make your own product that would be very similar to BAM, just using rice and milk, just basically produce a lactobacillus ferment. And I could send a website to Simone if she wants to see it. I have to be careful what I say because I can't, you know, but BAM is a good product, but you could also make your own, you know, like that's the thing. And it just depends how much of an area you want to treat, how much time you've got to make your own products. But there are a lot of, and in my opinion, of all the biostimulants I've seen, is that the ones that come from a fermented production system like BAM are actually the best, because all of your fermented products have much higher levels of microbial activity, and the microbes that are in those products, it's not the microbes themselves that do the have the beneficial effects when you apply them to soil, it's the biochemical signaling molecules that they produce which stimulates the seed. So that diagram I showed you of a newly germinating seed, and the microbes are moving out of the seed and they're sending signals back to the seed to tell the seed basically what kind of environment it's in, because the seed needs that information. If you apply a biostimulant to a seed or to an area around the plant roots, it's the signaling molecules that are in the biostimulant that the plant will respond to, not to the microbes, but to the signals they produce, because they're very similar to the signals that its own microbes produces, and it interprets those as coming from its own microbes and it responds to those. So it's like, you know, if a calf needs feeding and nudges its mum or something, or bellows to its mum will go, oh, calf needs feeding, so we'll produce more milk. Or in a human, you know, you get that let down response when a baby cries. So what's happening is that you're actually putting biochemical signals around roots or onto plants that the plant goes, oh, these microbes need to be fed, and will actually produce more exudates. So a biostimulant should stimulate root exudation in the plant, and those root exudates will actually feed its own microbes. So you're not adding microbes, you're adding signaling molecules of microbes. So a fermented environment is the one that will be very microbially rich and will have a lot of microbial signals in it. Now that fermented environment could be, like in New Zealand, we just saw there's a company there that produces fermented seaweed, which is an absolutely amazing product. You can produce something like effective microbes, which is a lactobacillus ferment, which is an amazing product. You can buy something like BAM, which is, you know, comes from a fermentation environment. It's an excellent product. So I would just give top marks to anything that comes from a fermentation background over an aerobic one.
Right. We've got a bit of an experiment running with some buried, a sack of buried rice that we're going to then dilute into a mixture and and experiment with.
Well, I don't know about that one, but you can make some very easily make some product just putting rice in water and waiting for it to form a film on the surface, and then putting some milk in it, and then the milk will separate into curds and whey, and your whey is basically pretty similar to BAM.
Okay, thank you.
Thanks. Thanks for your question. And great to hear you're doing some experimentation, because I think that's definitely something that I've taken away from your presentation, Christine, is just that need for farmers to be really doing like quite simple experiments on their own soil to get the evidence for themselves. I love that one that you showed with the 12 pots. And, you know, just that idea that we don't know what we don't know. And if we've been limited over time by thinking that there's only one way of doing things, it takes until someone opens our eyes to see there is in fact a whole lot more possibilities that we can be accessing. So yeah, that was a great experiment there, and so simple really when you look at it in the little pots there. So yeah.
What is interesting, Simone, is it's just on that note, when there's like been millions of dollars spent on research, and I've been involved in it myself when I worked at the university, where we did all these pot experiments in the glasshouse, and we always did them with monocultures. We never actually looked at diverse mixes, so that all the answers we got were actually not correct. And it's very difficult to get. We've got these deeply held beliefs about having to put things, like having to buy fertilizer and put it on plants. It's just so deeply ingrained, and it's because our whole culture has been based on that. So we have to start looking at things very differently and doing these little experiments, you know, to find that, well, there is a very different way of doing it, and understanding what the processes are. You know, if we don't understand what the processes are that we're trying to support, and the process you're trying to support is diversification of microbial activity in your soil. That's what you're trying to support. And the best way to do that is basically introducing a whole lot of different plants from different plant families.
Yeah, that's been a huge takeaway from your presentation today. And this group here, our mentoring group, actually had Colin Seiss talk to us a couple of months ago. So this has been a really fitting follow-on from that. So thank you. Thank you again for all you've shared with us today. And we look forward to keeping abreast of your work into the future too. Thank you.
Well, thank you, Simone, and thank you for the great questions. And I just, go forth and diversify. That's what I say. Okay. Good luck, everybody. Okay. Bye.
Thanks again, Christine.
Bye.
See you. Bye. Bye.
Thank you very much.
Thank you.